yes.
Great. I will present you an work which we done with our scientific group. The title is Anodal tDCS of the Broca's area improves learning of novel vocabulary. Last decade have seen a rapid rise of tDCS research of the human brain, where have shown a positive effect TCS of speech processes for healthy subjects and patients with speech disorders. TCS research on language processing has so far focused on the core language system of the health of the left hemisphere, including most importantly Wernicke's and Broca's areas. We already know that both anodal and cathodal tDCS of Wernicke's area in healthy subjects appear to facilitate semantic processing. Anodal tDCS of the Wernicke's area has also been found to facilitate associative learning as compared to sham stimulation, and anodal tDCS of the Broca's region improves both semantic and phonemic fluency. Furthermore, theory and her colleagues show that anodal tDCS of Broca's area improved speech repetition when Malutin and her colleague found that cathodal stimulation of the Broca's area increased naming speed. However, the field is still in its infancy and more research is needed to describe the effect of tDCS of verbal learning. Notably, the available results are mixed with respect of the specificity of stimulation polarity. Cathodal is anodal, and following the tradition in motor system neuroscience, most experiments have focused on anodal tDCS. Furthermore, no study has directly compared the effects of Broca with Wernicke stimulation on word learning in the same experimental design, what we had done. It is believed that word learning is realized via at least two mechanisms. One of them, often called explicit encoding, uses explicit direct instruction like he is turn reg or this is turn reg. It is associated with repetitive presentations occurring during practice session. Another one is fast mapping, FM, involves contextually driving implicit inference or deduction, usually based on exclusion, like does Tunrek has ears when you see Tunrek and Kitty. Most studies to date have focused on either one or the other learning strategy, with only a handful of reports comparing the two directly. Not all these have used tDCS to assess casually the relative input of different language cortex into these two mechanisms, since both learning types are important in natural language acquisition and word learning. The present experiment included in a counterbalance fashion both EE and FM condition. In our learning paradigm include an active semantic learning tasks for training new word form, which included two conditions aimed at acquiring a new word, either implicitly or explicitly. Each implicit or explicit condition include a picture of a target stimulus presented along with another image and was accompanied by an auditory presented question related to the picture. For our experiment, we used 16 real words, which mean in animals and objects, and we create 16 word form, all of which were tree fonts and fully conformed to Russian phonology and phonetics, but had no pre-existing meaning in the language. These 16 new forms were rotated across subject in such a way that they took on different meaning and learning role type in the learning procedure in order to rule out any idiosyncratic effects of always pairing the same word form with the same novel meaning. To compare visual stimulus materials corresponding to the spoken word form, we present real word unfamiliar for regular participants, such as ancient tools or musical instruments or deep sea animals. It were in monochrome color, 5 pictures for each object. And contextual equations for both conditions were composed in such a way that each trial had a unique combination of word form, picture, question. In total were the 10 trial per each new word for learning. And now we are going to procedure. One subject should learn just 8 new words, 4 words in EE condition and 4 words for FM condition. For the sake of clarify the role of the K language area in word acquisition with and without articulation task, for 15% of the tokens, we introduced a requirement to articulate word form at the end of the trial when prompted by a microphone icon. As a result, we used 2 stimuli of each type, which led to a 2 by 2 by 2 design. It's a learning type, animality, articulation. To each subject in a group, it should be 16 words in total, fully contrabalanced and rotated across subject. Response accuracies were assessed immediately after the trial session by free recall task. We quantified each answer as a number of correctly remembered phonemes for the newly learned word. And we use Anodal and cathodal tDCS of the 2 language area to access the effect of stimulation location and polarity of novel word acquisition. Sham, placebo stimulation, was used as a baseline condition. In a total, we had 160 healthy participants recruited by the study. The participants were randomly divided into 5 groups by 32 persons. You see 5 groups on the screen. Data analysis were done with using ANOVA by item and by participants Mann-Whitney and Wilcoxon tests. And we are going to the results. Participants correctly recalled an average near 15% of the new words after their learning task. Analysis of accuracy did not show a main effect of stimulation group by the participants or item analysis, but the total accuracy was significantly higher after Anodal Broca tDCS than after sham. Other groups, however, did not significantly differ from the sham baseline. We furthermore scrutinized the Anodal Broca condition further and planned comparison for the 2 learning regime indicated that response accuracy was significantly higher for Broca's Anodal than sham stimulation for both FM and EE condition. With respect to difference between the learning regime, also numerically of outcomes were better for EE condition than implicit condition. These differences were not significant. And finally, we focused on the articulation contrast. Wilcoxon test indicated that the subjects were better in remembering articulating words than non-articulating ones in both Anodal Broca groups and sham. An additional interaction between learning type, articulation, and stimulation group by participants or item. In accordance with the previous study, we found that Anodal tDCS other language-related areas improve in language performance, and we did not find the decreased performance of cathodal stimulation. But in our results, only Anodal tDCS of Broca's areas significantly improve implicit and explicit learning of novel vocabulary relative to sham stimulation. Yes, I'm finished. Thank you for your attention, and I'm open to your question.
Thank you very much, Ekaterina. Any questions? I'm trying to see if there are any hands.
Thank you. Very interesting talk. Did you also check for their associative memory? So you only tested, from what I could see, for their ability to recall, free recall as many words as they can. You're not sure if they associated them with the items that they appeared with, or did you test that, but you're not just not presenting?
We use different different tests, like lexical decision, semantic decision, and choose from 4. But in one presentation, I only discussed about free recall task and accuracy in this task.
And so just out of curiosity, so it looks like stimulation to Broca's area improves their ability to produce the word. Does it also improve their ability to make the association with the correct item? Because these might be, of course, different processes.
We try to answer this question. We, during all our experiments, can say that we always see common effects of stimulation, but specificity is in our discussed.
If I remember correctly, thank you for a very important question, Asaf. If I remember correctly, the free recall also showed effects of stimulation, but it was less specific. It was any stimulation, also cathodal, did improve memory.
Thank you.
So,
any more questions? I can see a question from Elizaveta.
Yeah. So it's technical question also. How did you choose the place to put in the tDCS? So was it just the projection of Broca area on the scalp or something like this?
We just use, like I show on the slide.
I missed it. Sorry.
Yes, it is an F5 and a CP5.
I see.
It's traditional for Brocas and Wernicke's area.
And what about the form of the magnet? Was it? Did it influence on the direction of the tDCS or not? So I've heard Professor Ilmaniemi, who said that these very complicated lines of the tDCS going through the brain, so it depends on the form of the magnet, on something like
it's not a magnet.
Yes.
It's not a magnet.
Oh, so sorry, not a magnet. Yeah, for sure. For sure. But what was the form of this tDCS?
I'm sorry, I couldn't understand the question in real.
It's a square shape electrode, if I remember correctly. And of course, it's not very precise. But the simulation suggests that it mostly goes through inferior frontal areas.
I see. Thank you.